My name is Janelle, and my project is on MIF Gene and Arthritis. An introduction. MIF, which stands for macrophage migration inhibitory factor, is a gene that has been linked to many diseases such as arthritis, lung cancer, kidney disease, etc. This project focused on analyzing the effect of MIF knockout in mice. This was done to find the top differentially expressed genes when comparing wild-type to MIF knockout. Since the MIF gene was commonly known to be linked to arthritis, an analysis of the CDC chronic disease indicator arthritis dataset was performed. This was done to find the prevalence of arthritis and to see if there is any correlation between the different indicators or questions. So, macrophage migration inhibitory factor, MIF, this gene encodes a lymphokine involved in cell-mediated immunity, immunoregulation, and inflammation. It plays a role in the regulation of macrophage function in host defense through the suppression of anti-inflammatory effects of glucocorticoids. It also has broad expression in kidneys, prostate, and 21 other tissues. Arthritis is the swelling of joints, mainly caused by an immune response. These are the datasets I used for the RNA-seq datasets. The samples I chose were the 07 and 08 for the MIF knockouts, and for the wild type I chose the 10 and 11. These samples were in the FASTQ format and they were peer-ended. For the arthritis dataset, I chose the CDC chronic disease indicator arthritis dataset. And the features I used were the year start, location, question, stratification 1, and data value, and also some IDs for those features. Built my hypothesis off of the different literature that I read. So the hypothesis for the MIF gene is that the top differentially expressed genes will have functions in immunological response. Her hypothesis for the arthritis set is that states with an overall large prevalence of arthritis will also have a large prevalence of patients who also have another disease, and there is also a larger prevalence in women compared to men. Okay, so for the methods and results, start with the methods. The initial analysis was done in the open source Galaxy website. So the first step was quality control, and I used FastQC for that. And then the second step, I used CutAdapt, but it wasn't necessary for this case because there were no major adapters showing up in the FastQC report. I just did it just in case if there were any other sequences in there that needed to be cut. And after that, I did alignment, and I used HESAT2 for that. And after that, I did differential gene expression. Then I created volcano plot and a heat map in R from the differential gene expression results. And I also did GoTerm enrichment analysis in R from the results as well. And go-term enrichment analysis is basically done to find the functions of genes. And then for the arthritis dataset, I did exploratory data analysis, and then I created pivot tables to get the prevalence values for each feature. Then I found statistics on the features based on the prevalence values, and I created heat maps to find the correlation between the questions based on the prevalence. This is the volcano plot created in R from the differential gene expression results. The plot shows the top differentially expressed genes in wild type versus MIF knockout. Most of the genes are downregulated in wild type, and MIF and FGF10 are upregulated in the wild type. From this graph, the ones in purple represent downregulated, ones in yellow represent upregulated, and gray is not significant. And then this table just shows the top 5 differentially expressed genes. Okay, so this is the heat map, also created from the results. And this heat map shows wild type versus MIF knockout. The blue represents downregulation and the red represents upregulation. So over here on the side where they're the most blue are the wild type samples, and on the side where they're most red are the MIF knockout samples. So the wild type samples had a lot of downregulated genes, while the MIF knockout had a lot of upregulated genes. Okay, so next I did principal component analysis to see how the samples separated. And most of the variation in the samples, which was around 90%, was covered by PC1. PC2, PC3, and PC4 were also able to separate the samples, but it was by less than 20%. So most of the variation in samples is between the conditions, so between MIF knockout and wild type, and there's very little variation in the samples for each condition. And the first graph just shows PC1 versus PC2. So this graph here. So you can see on the PC1 that the samples are separated by condition, and then PC2 separates the MIF samples, and then for this side you can see PC1 separating by condition again, but PC3 separates the wild type samples. And on the bottom you can see PC1 separating by condition, and PC4 separates both the MIF samples and the wild type samples, but the difference is very little. And as mentioned previously, go-term enrichment analysis is usually performed to look at the function of genes. The functions for the top 5 go-terms mostly dealt with mitosis, which is the process by which one cell divides into 2, and the graph on the side this one side shows the activated genes, and the other side shows the suppressed genes, and these are in wild type compared to the MIF knockouts. And you can see that most of the suppressed genes had to do with mitosis. These are suppressed in the wild type and activated in the MIF knockout. And most of the activated genes, their functions mostly dealt with sensory perception, air, morphogenesis, and cartilage, etc. And for this graph, the size of the dot represents the count, which is how many of these functions were present in the samples, and the color of the dot represents the adjusted p-value, how significant it is. Okay, so this slide is just the questions and the question IDs from the arthritis dataset table. So as you can see, some of the questions are arthritis among adults over 18, arthritis among adults over 18 who are obese, who have diabetes, who have heart disease. Then there's activity limitation due to arthritis, who have doctor-diagnosed arthritis, and there's severe joint pain, work limitation, physical inactivity, fear or poor health, and adults who have taken a class to learn how to manage arthritis symptoms. Results is based on the stratification feature, which mostly involves race and gender of the person. So on the stacked graphs, it's down here. Oops, down here. And it can be seen that the activity limitation had the highest prevalence, and according to the statistical table, the multiracial, non-Hispanic, and Black non-Hispanic had the largest average prevalence values of around 40 to 44, and male and white non-Hispanic had the lowest values of around 32 to 35. So I did the same thing with the location feature, and based on the stacked graph, arthritis with heart disease had the highest prevalence, and based on the statistics table, Hawaii and the Virgin Islands had the lowest average value of around 24 to 29, and West Virginia and Kentucky had the highest values of around 45, and by value I mean prevalence. And then next I did the same thing with the year start feature. I just wanted to see if there was any new information I can uncover, like if there were any changes throughout the years or anything. And arthritis had the highest, arthritis with heart disease had the highest prevalence, based on the stack graph, and then based on the statistics table, 2020 had the lowest average value of approximately 34, and 2015 had the largest average value of around 37. So it's not a huge difference, but yeah. Next, I did heat maps of the stratification, location, and your start prevalence values. And the correlation is between the questions based on these values. So there was a high correlation between the people who had arthritis along with heart disease, diabetes, and obesity. There was also a correlation between activity limitations, work limitations, fear, or poor health, and adults who have taken a class to learn how to manage symptoms. This probably means that the people who are severely impacted but can still move but are like limited are more likely to take these classes. And physical inactivity had a negative and low correlation with people who took these classes. So this means that people who are inactive are less likely to take these classes, maybe because their situation is extremely severe and requires another type of treatment. Conclusion: the results of the differential gene expression of wild-type versus MIF knockout show that more viral response genes are activated in MIF knockout along with the cell development genes. The analysis of the arthritis dataset shows that men are less likely to have arthritis than women, which supports previous literature. Also, the states that had a high prevalence of arthritis had the highest amount of arthritis, with heart disease and diabetes. And this shows that a reduction in these diseases can help in reducing arthritis. Also to note, there was high prevalence of arthritis in certain races, but I think more specific look into which type of arthritis needs to be done, because according to previous literature, the results aren't very consistent. So I think a more specific arthritis problem can help narrow down the accuracy of the results. And also the top 5 differentially expressed genes have immunology functions, so the null hypothesis of the MIF gene analysis is rejected, and since the results for the arthritis dataset align with previous literature, most of the results did. The null hypothesis of the arthritis analysis is also rejected. A limitation of the study was not having access to MIF knockout datasets in arthritis patients, along with that dataset being large enough to perform predictive analysis. Also, there were not many publicly available arthritis datasets. For future work, the MIF gene in arthritis patients versus MIF knockout or MIF inhibition will be interesting to study so that a better understanding of the mechanism can be achieved. This can possibly help in developing treatment options. For references, and if you have any questions, let me know. Thank you.